Today we are starting a series of lectures on a very basic topic in the medical sciences, that is the physical structure of a cell, right? First of all, I will discuss about the structure and function of the cell membrane. Cell membrane is also called plasma membrane of a cell, right? So let's talk about the structure of a cell membrane. Let's suppose here is a cell. Suppose we take a typical cell here, right? Now all of you know that cell has a basic structure. It has a nucleus and there is cytoplasm and it is surrounded by the cell membrane, right? Now we will go into detail of the structure of the cell membrane. Primarily, cell membrane is made of phospholipids and associated proteins. Again, what are the basic two components of cell membrane? This is made of phospholipids and associated proteins. Is that right? Now, let me draw this structure right in detail. Basically, this cell membrane is a double layer of the molecules. Primarily, it is a double layer of molecule. Because it is double layer of molecules, so it is also called lipid bilayer. Because it has a lot of lipid and it's a double layer of the molecule, it is also called lipid bilayer. Now, let me draw a component of it. Suppose this is a cell and I just draw a cell membrane in detail from this point up to this point only, right? First of all, the basic component is phospholipid. Now phospholipids are what? Phospholipids are having one part which is polar, right? This is the polar part of the phospholipid. Here there is phosphate and with that it is having fatty acids. What is it? Fatty acid. This is the basic molecule which is going to make cell membrane. So in the cell membrane there are phospholipid and it has one polar compound, polar end, and other are fatty acids, right? Now we have to see how a lot of these molecules make the cell membrane. Before we go how these molecules are oriented, you must know that extracellular environment is aqueous environment. There's a lot of water outside the cell membrane, and there's a lot of water inside the cell membrane. So it means extracellular environment is aqueous environment, and intracellular environment is also aqueous. Is that right? There's water outside the cell and there's a lot of water inside the cell. So it means this cell membrane is separating extracellular aqueous environment from the intracellular aqueous environment. Is that right? Now, you must be knowing that polar compounds dissolve into polar compound, right? This is polar component of the phospholipid. Because this is polar, it has some charge. It is water soluble end of the molecule. And these two fatty acids, these are not water soluble. These fatty acids are not water soluble. This component of the molecule which is water soluble, it is called hydrophilic. What is this component called? Hydrophilic. And this component which does not dissolve in water, this is called hydrophobic. So we can say phospholipid molecule have hydrophilic component. They have a head which is hydrophilic and they are having tails which are hydrophobic. Is that right? Now, what really happens that in this part of the membrane, this water soluble part will be oriented outward towards the external environment, right? And what are here? These are, yes please, fatty acids, and these are hydrophobic. Is that right? Now, actually this is one layer. Other layer will be on this side, layer of the molecules. Now, you can see that cell membrane is made of double layer of phospholipids, right? I have shown you the molecular structure of the cell membrane. Now, question is that why these molecules of phospholipid are oriented in such a fashion? Answer is very simple. You know, these components, fatty acids, they are not going to dissolve into water. They avoid water. They are hydrophobic. So it means there is water outside and water inside. So outer layer of the molecule, this hydrophobic component will be directed away from the water, and in inner layer again these fatty acids will be directed away from the water. So they will be directed towards each other. Is that right? So what we can really see that fatty acids, right, they dissolve into each other, they attach with each other, they are oriented towards each other. Just like you put a drop of oil and another drop of oil in the water, oil drops will fuse with each other. You take a little bit of water, put 2 drops of oils. Eventually what will happen to those drops of oils? Oil dissolve into oil. Is that right? In the same way, there is water outside, water inside, and this is something like oil, fatty acids. So they are oriented towards each other and they make the central portion of the cell membrane. But as you know, these blue parts, right, of the phospholipids, this is charged part or polar part or hydrophilic part. Hydrophilic means water loving part. This component is also water loving. So fatty acids which are in the outer component, they are, what is this, hydrophilic part will be oriented outward, and phospholipid which are inside, inner side of the membrane, their hydrophilic component will be directed towards the inner side. So what really happened, that cell membranes are made of double layers of phospholipids, and phospholipid molecules are oriented in such a way that in each layer fatty acids are directed towards each other and hydrophilic polar components of phospholipids are directed towards the external environment, aqueous environment, or they are oriented towards the internal aqueous environment. Am I clear? This is the very basic structure. We have not talked about the associated proteins. Now another thing. You see, the core of the cell membrane is having a lot of lipid inside. What is this? This is all lipid. So it means those compounds which are highly charged or those compounds which are very large, can they directly enter into cell? No, because only those things can cross this membrane which are lipid soluble. You know, core of the membrane is made by the lipid. Is that right? So if there is some substance here which is lipid soluble, it can dissolve into membrane and come in. Or there is any other substance which is lipid soluble inside, it can dissolve into membrane and go out, right? So which substances can cross the cell membrane easily? Which substances can cross the cell membrane easily? Substances which are lipid soluble and substances which have very small molecular weight, right? So we can say that substances which are lipid soluble or substances with very, very small, yes, molecular weight, these substances can directly go into the cell, or if these substances are inside, they can easily go out. Why? Because they can dissolve into lipid and pass through that. But other substances which are larger molecules, or those substances which are highly polar. Polar substances mean which are having charges on them. For example, this is another substance, and this substance or molecule is having charges on it. It's a polar compound. Can it dissolve into lipid? No. So can it enter directly into cell? Can it enter? No. So it will be repelled away. In the same way, let's suppose this is another compound, and this is very, very large compound. If it's very large compound, can it dissolve into lipid? No. And can it go out? No. What does it mean then? It means that this lipid component of the membrane is providing the permeability barrier for the large compounds and for the highly polar compounds, that they cannot pass through membrane. It means if some very large molecule has to come in, or any large molecule or polar or charged molecule has to move through the membrane, it has to have some special facility to pass through the membrane. Let me repeat it. I just said up to now that cell membrane is made of lipid bilayer primarily and associated proteins, right? Proteins I did not discuss up to now. And in lipid bilayer, what is happening? Mainly it is consisting of phospholipids. Is that right? And phospholipids are having the polar heads and hydrophobic tails. Hydrophobic tails of fatty acids are oriented towards each other, and polar heads of the molecule are either outside towards extracellular aqueous environment or oriented inside towards the intracellular aqueous environment. So core of the cell membrane is made of primarily lipids, right? And those substances which are lipid soluble or very small molecular weight, they can easily pass through the cell membrane. But other substances which are very large molecule or which are having a lot of charge on the molecule, they cannot pass through the membrane by simple diffusion. Is that right? It means it's for such substances, if they have to pass through the membrane, we need special arrangement. There should be special windows and there should be doors through which these membrane can slip in or out. Those special windows or doors within the cell membrane are made of which component? Proteins. In the cell membrane, there are special type of proteins. Is that right? And those proteins, for example, let me tell you here that here is a protein, and let's suppose, this is a protein molecule made of multiple peptide chains, and these peptide chains, suppose this is one peptide chain, this is second, this is third, this is fourth, right? And these peptide chains are arranged in such a way that in the center of this molecule, right, there is a longitudinal tunnel. There is a tunnel, and this is water-filled tunnel. What is it? Water-filled tunnel. We call it aqueous channel. What is this channel? This channel has a hole which is full of water, and then those substances which are water soluble, they can pass through this. So this is one way, one of the function of the proteins, that proteins are present in association with the cell membrane phospholipid so that substances are allowed to pass in and out. Such type of proteins which are present in the cell membrane, they are called channels. What are these called? Channels. And I will tell you the functions of proteins, more functions of proteins later. Let's come back to this component. After that I will go into detail of the protein. Another thing, the types of fatty acids and phospholipids which are in outer leaflet and which are in the inner leaflet, they are not identical. I will go to the proteins in detail later. Again, listen. You know, this is the outer leaflet of the cell membrane and this is the inner leaflet of the cell membrane. Now, these phospholipids which are present in outer leaflet are slightly different than the phospholipids which are present in inner leaflet. It means phospholipids of outer leaflet and inner leaflet are not identical. So it means outer leaflet phospholipid structure and inner leaflet phospholipid structure is not symmetrical. They are having asymmetry. What we call this term? Asymmetry. Now, which are the phospholipids which are present in outer part of the cell membrane? Number one, these are phosphatidylcholine. Okay, I will write it before you start crying. Phosphatidylcholine. This is a type of phospholipid, right? Phosphatidylcholine. This is present abundantly in the outer leaflet, right? With it, there is another friend phospholipid which is called sphingomyelin. Sphingomyelin. So, phosphatidylcholine and sphingomyelin are two types of special phospholipids which are abundantly present in the outer leaflet of the cell membrane. Right? These are the two phospholipids which are present in the, yes please, outer leaflet. Now we'll talk about what? Phospholipids which are specially present in inner leaflet. Right? Again, there are two important phospholipids which are abundantly present in the inner leaflet of the cell membrane. One is phosphatidylethanolamine. I don't know who was man who was putting the names of these. You could put simple name like Carla, Joanne, I don't know. He's sick man who put these names. Right? So, the inner part of the cell membrane, it is having some funny type of, you can say phospholipid, phosphatidylethanolamine. Phosphatidyl, okay, ethanolamine, right? This is one. And the second type of phospholipid which is abundantly present in inner part of the inner leaflet of the cell membrane, that is called phosphatidylserine. Phosphatidylserine, right? So because phospholipid in outer part are somewhat different and phospholipids which are present in inner part of the membrane, they are slightly different. So we can say that cell membrane, outer leaflet and inner leaflet show some degree of asymmetry. They are showing some degree of asymmetry, right? Later on we will learn that another component add to the asymmetry, that the proteins which are attached to the outer part are slightly different than the proteins which are attached to the inner part. So not only difference of phospholipid make the asymmetrical situation, later on we will learn the proteins which are attached to the outer part of the leaflet and the proteins which are attached to the inner part of the leaflet, they are slightly different from each other, so they also contribute to the asymmetric situation. And still later on we will learn that outer leaflet has a lot of carbohydrates attached with it, but inner leaflet does not have any significant carbohydrate. So it means outer leaflet is having a lot of carbohydrate molecules outside, inner leaflet does not have carbohydrate molecules. Again, this difference of carbohydrates applied on these leaflets also adds to the asymmetry of the inner and outer leaflet of cell membranes. I think I need to recap this. I was trying to put a concept in your mind that cell membrane is made of phospholipid bilayer. with associated proteins. Is that right? And it is making outer leaflet and inner leaflet. And outer leaflet and inner leaflet are not identical. If they were identical, we could say that they are symmetrical, but they are not symmetrical. Why they are not symmetrical? There are multiple reasons. One reason is that phospholipid in outer leaflet are different and phospholipids in inner leaflets are different. Second reason, the proteins associated with outer leaflet are different and proteins associated with inner leaflet are different. Third reason, outer leaflet has lot of carbohydrates attached outside, but inner leaflet does not have any carbohydrates attached inside. So all these reasons make the outer and the inner leaflet of the cell membrane asymmetrical or non-identical to each other. Am I really clear? Anyone who does not understand, raise your hand, I will run away. Everyone understand, that's good, we can continue. Another thing, in between the legs of these fatty acids, there is some naughty molecule here. It loves to be here. I don't know why. Who knows this naughty molecule? Yeah. Anyone, please use your own head. This is a very special type of lipid. Okay, let me tell you. I will draw that structure in a large way. Right, and there are their beautiful legs. Okay, it's a little bit, I should say, having a dancing tendency. Now, yeah, what are these? These are polar ends, right? Water soluble ends, and these are lipid soluble. And there are some molecules which love to be here, naughty type. Who will tell me what are these molecules? You know it. I'm sure you know the name of these molecules. We call them cholesterol. Have you heard of cholesterol? So cholesterol is another type of lipid which is present in the cell membrane, intercalated in between the legs of the phospholipids. Is that right? And cholesterol molecule have hydroxyl. This is a hydroxyl ends are oriented outward. Why? Hydroxyl are polar end. So most of the cholesterol molecule is lipid soluble. So it is oriented inward. But their hydroxyl ends which are polar or charged, they are oriented outward. Is that right? So we have learned that cell membrane most important lipids are phospholipid, but they do have a small percentage of cholesterol molecules intercalated in between the phospholipids. Is that right? Any question up to this? There is no. Another thing, very interesting. These phospholipid molecules are really not fixed. They are not fixed. For example, this molecule is present here. You look around and that may slip on other side. What does it mean? All these molecules are mobile. Phospholipid molecules are mobile. They keep on moving laterally. Is that right? They are not fixated in their position. These phospholipid molecules are not fixated in their position and they display the lateral movement. And this concept of lateral movement is called fluidity of the membrane. The membrane molecules are like a fluid, and in a fluid area they are mobile. They are not fixed in a particular specific position. Is that right? So phospholipid molecule along with cholesterol, they can slip laterally, right? And we say that membrane shows the property of fluidity. Property of, yes, fluidity. This is an important property, fluidity. You know why it is so important to know that membranes have some fluidity, because later on you will learn, right? Sometimes cell has synthesized something. Let's suppose cell has synthesized a hormone. Now hormone molecules are packed into a membrane bound vesicle. This vesicle may come and fuse with the membrane. It may fuse with the membrane, and these substances may go out. Many cells secrete substances. For example, cells in the pancreas, beta cell of the pancreas secrete insulin. So if it is a pancreatic cell, there will be insulin containing vesicle, and these vesicles are membrane bound. And when insulin is going to be released, this vesicle will move towards the membrane, and membrane of the vesicle will fuse with the cell membrane. And where they fuse, a small puncture point is made, and insulin will go out. And then due to fluidity, it will seal. It will be completely closed. Is that right? If this was a fixed membrane, can it easily go out? No. So this process is called exocytosis. The cell is showing exocytosis. So if fluidity is important, so that cell can show exocytosis. Sometimes cells are in a mood to eat something. Let's suppose here happen to be a bacteria. Right? This is very sad. You know why? Because cell is going to eat this bacteria. If this is a white blood cell, now this is the membrane of the white blood cell, right? And it has catch the bacteria, and then this membrane will progressively move to each other, and then eventually it will fuse, and this will go inside. This process is called endocytosis. What is it called? Endocytosis. Again, because membranes are fluid, so during the endocytosis, a piece of membrane has invaginated and then pinched off, right? Still there is no hole in the membrane. Or will there be any hole in the membrane? There will be no hole, because as soon as it remove away, phospholipid come on the side and seal it. Is that right? In the same way, when this membrane from inside going out, these phospholipids are added to the membrane. So for the process of exocytosis or for the process of endocytosis, membranes must have some degree of fluidity, right? And then this type of, fluidity is also important for membrane biogenesis. Biogenesis means membrane synthesis. When cells are initially small, when cells divide, daughter cells are small. With the time, cells keep on growing. So it means their membranes should also grow. How the membranes grow? Cells synthesize new phospholipids and keep on inserting in the membrane. And because membranes are fluid, so keep on accommodating the new molecules and enlarging their surface area. So it means fluidity is important for exocytosis, it is important for endocytosis, it is important for membrane biogenesis, biogenesis, and fluidity is also, right? These are some important reasons why fluidity is important. Now, what are the factors which can alter the fluidity of the membrane? For example, if phospholipid molecules can more freely move, we say membrane is more fluid. And if the movement is relatively less free, we say membrane is less fluid. Now, do you know any factor which can alter fluidity? Temperature. If temperature goes up, membrane will become less fluid or more fluid? Of course, more fluid. There is no need to explain it. All of you must know. Temperature increases the movement of the molecules, and molecules which are already ready to dance, they will dance more when temperature of the membrane goes up. So this is very easy, no need to remember it even. That increasing temperature increases the fluidity of the membrane. Now another thing. Increasing the cholesterol content in the membrane. If we add more and more cholesterol in the membrane, fluidity will increase or decrease? Yes, very good. How you know that? Yeah, that's right. He knows so well that when you are adding the cholesterol, actually, their legs cannot move now. They're packed. Membrane components are more tightly packed. When you insert the cholesterol, you intercalate the cholesterol molecule in between the phospholipid molecules, and membrane become less fluid. So you can say adding the cholesterol make the membranes less fluid. Another thing, these are the straight fatty acid, and this is some, what's wrong with this fatty acid? Yeah, what is wrong with this fatty acid? It is having a kink in its leg. Yeah. Can't you see it? You don't need to call, I think, Aristotle to discover this. It is not straight, it is having a bend in it. Why it is having a bend? Some fracture there or what? Actually, when fatty acids are saturated, they are straight. When they are unsaturated, they have a kink. You will study in biochemistry one day, right? That when fatty acids are fully saturated, they are straight, relatively straight chains. And when fatty acids are unsaturated or relatively unsaturated, they have bend in their structure, not in their leg. Who told you they have legs and shoes? No. Right? Now what is there? They have a kink. Now what do you think? If in the membrane most of the fatty acids are unsaturated, it means they are having bend. This will be more fluid or less fluid? Yeah, it will be more. Someone moving leg like this and this, people tend to move away or move in laterally. So what really happens? When fatty acids are relatively unsaturated, membranes are more fluid. And when you make the fatty acid more saturated and all fatty acid become straight, they become tightly packed, membranes are less fluid. Am I clear? No problem up to this? Right? We have just talked about the fatty acids. Their cell membranes are made of by double layer of phospholipids. Is that right? And a polar ends of the phospholipids are oriented either outward or inward, and hydrophobic fatty ends are oriented to each other. Is that right? Then what did we discuss? That these membranes are symmetrical or asymmetrical? They are asymmetrical because type of fatty acid in outer leaflet are different than the type of fatty acid in the inner leaflet. Plus proteins outside and inside are different. Thirdly, outer side has a lot of carbohydrate, inner side does not. Is that right? Then we talked about fluidity, that phospholipid molecules can float around in the membrane. So we say membranes show the characteristics of fluidity. And why fluidity is important? Because there is processes of exocytosis going on, there are phenomena of endocytosis going on in the membranes, and then there's membrane biogenesis going on, right? So due to all these reasons, it's very important that membrane should be appropriately fluid. Is that right? So that there are no fractures and punctures in the membrane. Is that right? They should seal off. Then we talked about that fluidity can be increased by temperature, but decreased by adding cholesterol or making the fatty acids more saturated. Is that right? Yeah, that will make less fluid. If it becomes more saturated, membrane is less fluid. If there's more cholesterol added, membranes are less fluid. Then another thing. Okay. Let's suppose this is a container with lot of milk. There's a lot of milk inside, right? And there is a butter. Butter float into, butter, right? And you imagine, here's a frog sitting. Can you imagine? I don't know how to make a frog, but anyway, this is a frog. Something like this is also present in the cell membranes. There's a frog sitting on the butter and using that piece of butter as a personal raft to move and float on the milk. Is that right? Something like that apparatus is also present here. Let me tell you what is that apparatus. Of course, there is no frog there. Actually, in some part of the cell membrane, what really happens that phospholipids are more tightly packed, and not only phospholipids are more tightly packed, their fatty acid components are longer. The fatty acid components are longer. And they are very tightly, what is this? Cholesterol. So in some part of the cell membrane, there are some specialized structure like butter piece here, right? And then they become normal structure. Now in this specialized structure, what is happening, that fatty acids are more saturated and they are longer, and this area is having unusually high concentration of cholesterol, right? This is like butter piece, and this butter piece is moving in the membrane. Membrane is like milk layer, and this butter piece is moving. And what is the frog? Frog are special type of proteins present over there, right? For example, there may be very special protein present over here. What is this? Can you tell me what is this? In your medical studies, you will come across something like this again and again. These are special type of receptor proteins. Now let me tell you what is receptor. Receptors are special macromolecules which will bind with a, okay, let me make a simple receptor. Let's suppose this is a protein. Now listen, this is a protein, and when this protein binds with a special type of substance, protein undergoes change, and this component of protein alter. Again listen, if I'm a receptor, I have one end outside the membrane. Another end inside the membrane. Is that right? If you put some piece of gold in my hand, I start telling you, yes, I have received it. What does it mean? That I can bind the gold outside and I can give signal of reception inside. Is that right? So what I'm doing, I'm a molecule. On one side, I can bind a specific substance. On other side, I can give the signals and modify the biological system. Such molecules which can bind with one specific substance and then produce modification in the biological system, such molecules are called receptors. So what are receptors? Receptors are macromolecules to which, when a specific substance binds, that specific substance may be hormone, or that may be neurotransmitter, or that may be a drug. So anything specific bind with the receptor, receptor will give signal to the biological system and alter the function of the biological system. Is that right? Now, actually, what is this? This is also a receptor system. This is a protein, this black system is a protein or peptide chain which is acting as a receptor. Why I say it is acting as a receptor? Because here it will receive the stimulus. Whenever this molecule bind here, this start moving the tail end, and this will give signal into cell. For example, this is epinephrine or norepinephrine. This is the receptor of epinephrine or norepinephrine. When epinephrine bind here, The receptors give intracellular signal. Why? Because epinephrine cannot enter into cell. Epinephrine is highly polar compound, it cannot enter into cell. So extracellular stimulus can bring intracellular response due to the presence of receptors in the cell membrane. Again, let me tell you. In the cell membrane, attention please. You know, someone comes to visit you. If he is decent enough and if he is not allowed to enter your home blindly, he should stand outside like a thorough gentleman outside your door, and then he should push the what? Doorbell, and then there will be tring, 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 and inside your house some activity may change and you may rush to the door. So what is doorbell? Doorbell is a receptor. Doorbell is a receptor. That, and who was the person who came and pushed the doorbell? He was the neurotransmitter or hormone or drug. When he binds with the receptor, it gives signal inside the cell. Is that right? So what are receptors? Receptors are macromolecules, usually proteins, to which a specific substance binds and bring the intracellular changes or bring the changes in the biological system. Am I clear? Now let me tell you something interesting. Some substances are lipid soluble and they can go into cell. If a substance can go into a cell, its receptor should be present within the cell. Let me tell you. Let's suppose this cell is under the influence of two hormones. One hormone molecule is thyroxine. Have you heard of thyroxine? Thyroxine. Thyroxine is a very, very small molecule and it can easily go into the cell. Right? Another hormone is insulin. Let's suppose another hormone is insulin. Insulin is a very large molecule with a lot of amino acids. Now, thyroxine can go into cell. Insulin cannot enter into cell. So, receptor for thyroxine should be present inside the cell, and receptor for insulin should be present on the cell membrane. Am I clear? So, what I am really trying to tell you? I am trying to tell you all those substances which cannot enter into cell. For those substances, receptors should be expressed on the cell membrane. What is the function of the receptors? Membrane receptors, they take the extracellular signal and produce intracellular responses. Is that right? Am I clear? No problem? Now, of course, whenever you see there is a hormone which is very large or highly polar, its receptors should be on the surface. Let's go back to our frog. We were talking about that in some specialized area of the cell membranes become little thick, phospholipids and cholesterol tightly packed, and lipid fatty acids are very long and fully saturated. Is that right? And they are having special type of proteins attached with them. And these proteins which are fixated on this act like a frog. They are riding over it and riding through it. What are these? These are receptors. And there may be some receptor associated proteins also. Maybe this is the receptor associated proteins. Now, hold this signal mechanism, hold this signaling proteins along with this structure are called lipid rafts. What are they called? Lipid raft. You know, rafting. Is that right? So we say receptors and their associated proteins are rafting on the what? In the membrane in specialized area. Am I clear? No problem up to this? Right. So what we have learned up to now, that we have talked about the phospholipid nature of the cell membrane, and we have talked about that there are lipid rafts present. We have talked about the fluidity and the factors which affect the fluidity. Is that right? We have a break now, then we'll talk about the proteins associated with the cell membrane. Right, in the very beginning of the lecture, we said that cell membranes are made of phospholipids, cholesterol, and associated proteins. Let's discuss associated proteins in little bit detail. Actually, all the proteins which are associated with the cell membranes can be primarily divided into two types of protein. One type of the proteins which pass through and through the membrane. For example, this is a protein which is passing throughout the membrane, right? And there may be some proteins which are just you can say attached outside the membrane, or they are attached inside the membrane, or these proteins may be hooked with the now look, this red color protein is a protein which is passing through and through the membrane, and these green proteins are either present on internal side of the membrane or attached on the external side of the membrane, right? Those proteins which pass throughout the membrane, these proteins are called integral proteins. What are they called? Integral proteins. So membranes have special type of integral proteins which are passing through the membrane, and these integral proteins are also called transmembrane proteins. What are they called? Transmembrane proteins. So integral proteins, the transmembrane proteins are those proteins which pass through and through the membrane, and they do have an extracellular domain as well as they have an intracellular domain. Is that right? So naturally, and what about these proteins? The green proteins, they are not passing through and through the membrane. They are expressed either on external side of the membrane or they are expressed to the internal side of the membrane, and these proteins are called simply peripheral proteins because they are on the peripheral sides of the membrane. So they are called, yes please, peripheral proteins. So peripheral proteins are loosely attached with the membrane. They are loosely attached with the membrane, and they may be attached on external side of the membrane or internal side of the membrane, and these peripheral proteins are attached with the membrane in a non-covalent ways. They are not covalently attached, because if they are attached covalently, then they will be strongly attached, but they are non-covalently attached with the membrane, and they can be easily shaken away from the membrane. They can be easily removed from the membrane. Just changing in the ionic composition or changing the pH, you can remove the peripheral proteins. But integral proteins are really the part of the membrane. Integral proteins, because they pass through and through the membrane, so if you really want to remove the integral protein, you need to disrupt the membrane. Am I clear? Yes. No problem. Now, I will go into detail of integral proteins first of all. Before I move forward, the total proteins associated with the membranes make about 50% of the membrane component. So you can say that cell membranes are 50% proteins, and remaining 50% has about 47% lipids and 3% carbohydrates. Are you understanding me? You don't understand me, okay. A cell membrane has proteins how much? 50%, right? And lipids are how much? About 47%, mostly phospholipid with some cholesterol and other lipids. And then there is carbohydrate. Carbohydrates are only 3% with the membrane. right? We have discussed about the lipids in detail. Now we are going to talk about proteins, and then we will talk about carbohydrates. About the proteins, I am saying there are two types of proteins. There are integral proteins and there are peripheral proteins. Is that right? Now, I will go into detail of integral proteins, right? What are the functions of integral proteins? Why integral proteins have to be there in the membrane? They are a very, very important function. Number one, as I told you in the beginning, that those substances which are highly polar or very large substances, for example, there is a polar compound, even if it is small, you know, sodium, it is a charge, or calcium, it is having charge, or chloride, it is having charge. These substances cannot diffuse through the membrane, or can they diffuse through the membrane? Or even potassium, it is having a charge on it, it is not lipid soluble. Can it diffuse out through membrane? No. But when we need to move such substances which are not lipid soluble, but we need to move through the membrane, then we need to take a help of a protein. How the proteins can help us to move the substances through the membrane? There are two ways. One way is that proteins make channels. As I told you previously, that let's suppose this is one peptide chain. Okay. Let's suppose this is one peptide chain. Then I make here another peptide chain. Then there is here another peptide chain. And in the end, another peptide chain. Let's suppose how many peptide chains I have made here? 1, 2, 3, 4, 5. These are 5 amino acid chains or peptide chains. If you put them together, they will make a channel. They will make a channel. And such channels are present in the cell membranes. These channels are made of what? Carbohydrate, proteins or lipids? Yes, please. These channels are made of proteins, amino acids. And in this channel, this central area is full of what? Water. This is having water filled central tunnel. We call it aqueous tunnel. The water can move through and through from this. So many water soluble substances can pass through the channels. Is that right? So what did I tell you? That one type of integral protein is, a whole group of integral protein is working as channels. And these channels can help the substances to move across the membrane. This is one type of integral, one function of integral protein. Then another integral protein. I don't know how to make bird, but some proteins are. This is another integral protein which is present through and through what? Membrane. What it is doing? It can catch a substance here. It will catch that substance here, and as soon as a substance binds with it, it moves through that and releases the substance in. Let me show then what it will do. So what has happened to this protein? This protein has altered its structure. Such alteration in protein structure is called, transformation. That some proteins undergo some transformation when they bind with a substance, right? And the mouth of this protein will take the substance, carry the substance inside. Such proteins are not having the central water channel. So these are not channels. These are simply called as a group. These proteins are called carrier protein. They carry a substance, undergo some change, and shift the substance across the membrane. Is that right? But they are very specific. Some carriers are only for one substance. There are other carriers which are only for other substances. They are specific what they will transfer. So this is second type of integral protein. So some integral proteins are working as channels. The other integral proteins which are working as carrier proteins. They are working as, yes please, carrier proteins. You understand why these proteins must be present in the membrane? Because cell has to take a lot of substances inside, or whatever it has synthesized, it may need to release them out. Is that right? So these are carriers, carrier proteins, right? Then there is another group of course, this beautiful design should be there also. Okay. So these were channels, these are carrier proteins. Then there are some other proteins also which are present throughout the membrane. Right? And these proteins, even though they are through and through the membrane, they can get a substance, bind a substance A plus B with them, and then release them as C plus D. What is this protein? Enzyme. You know, this is the metabolic function of enzyme. They can take one substance and convert into another substance, or enzymes facilitate biochemical reaction. So there are some enzymes which are present within the within the cell membranes, right? Of course, some enzymes are attached outside or inside. If enzymes are attached on the external aspect of the membrane or internal aspect of the membrane, then enzymes are there as peripheral protein. But some enzymes are present through and through the membrane, and then they are considered as component of integral proteins. So what we are talking about, membranes are not just simple biological barriers. They are dynamic functionally, right? They are having channels, they are having transporter carriers, carrier proteins, they are having enzymes as integral proteins, right? What else membranes can have? Anyone who can tell me? They are having special linker proteins. Look at this monkey. This is another integral protein. Its hands are dangling out and legs are dangling in. And what it is doing there? Outside, maybe this protein is holding some special type of, suppose, collagen. Do you know collagen? Collagen is a protein which is present outside the cells. And inside, there may be actins. It is sticky to some filaments called actin. So what it is doing? It is attaching this part of the cell with extracellular environment. But it is just structurally fixating it. You are understanding it? What is the advantage of this? That this is a molecule which is molecule of a protein which is transmembrane, and it binds the internal cytoskeleton with extracellular component and help the cell for its structural stability. Are you understanding? Different cells and different tissues need to be structurally attached, right? And different cells have different shape. Now, this is intracellular proteins which are also called cytoskeleton. As you have bones, you know, you are having bones, I think you are sure. Bones help you in many things, including giving you a shape and strength. Is that right? In the same way, as you have skeleton, cells also have skeleton. These are skeletal proteins of the cells As a group, these proteins are called cytoskeleton. So cell membrane inside has cytoskeleton. There are many cytoskeleton proteins, and outside there are extracellular collagen and other proteins, right? To stabilize the internal cytoskeleton with external extracellular matrix, we use the term for this, extracellular matrix. We need some types of proteins which are what? Transmembrane, integral. On one side they can hold the extracellular components, in other side they should be able to hold the intracellular cytoskeleton. And these proteins are also example of what type of proteins? Yes, what type of proteins? Integral proteins. Is that right? So how many functions of integral proteins you have seen? Integral proteins may be acting as channels, they may be acting as carrier proteins, they may be acting as enzymes, and of course they may be acting as, what is this? Linker protein. They are producing a link between intracellular protein and extracellular proteins, linker proteins, right? Linkers protein. Then integral proteins have one very important function, one more. And what is that? Okay, let's suppose this is a protein which is present throughout the membrane, and this protein has extracellular component, it has transmembrane component and intracellular component. Is that right? Let us suppose this protein can bind the insulin. When insulin comes, insulin binds over here. When insulin binds over here, right, it starts giving signals inside. So this protein is working as receptor. So receptors in the membranes are also integral proteins. So today onward, remember, cell membranes are made of phospholipids and associated proteins. Associated proteins may be integral proteins or peripheral proteins. Integral proteins are transmembrane protein or through and through proteins. Integral proteins have many functions. They may be functioning as channels, they may be functioning as carrier proteins, they may be functioning as enzymes, they may be functioning as linker proteins, they may be functioning as receptors. Is that right? Broad main categories of, functional categories of integral proteins. And about peripheral proteins. Peripheral proteins, as I told you, they are attached to the external aspect of the cell membrane or internal aspect of the cell membrane. And peripheral proteins are loosely attached or non-covalently attached, and they are held with the membrane only with the weak, very weak electrostatic forces. So they can be disrupted very easily away from the membrane, the peripheral proteins, right? And peripheral proteins are basically functionally acting as cytoskeleton. Sometimes what happens, look here, inside the cell, at this point, some proteins are attached like this, right? Or here are some proteins, and they are stabilizing the structures of the cell. Now these protein filaments are attached with the internal aspect of the cell, right? So they are transmembrane, or they are integral, or they are peripheral proteins. They are peripheral proteins. So many of the peripheral proteins which are attached to the inner aspect of the membrane, they are component of the cytoskeleton. They are the component of the cytoskeleton. Even though I told you cytoskeleton protein, which is a peripheral protein, may be associated with linker protein. They may be associated with linker protein, which may link the cytoskeletal peripheral protein with extracellular matrix. Am I clear? Then another important relationship or function of the peripheral protein is. Listen, some of the peripheral protein are working with the receptor. Let me tell you how. Let's suppose here is a receptor, and this receptor protein passes 7 times through the membrane. Such receptor which peptide chain passes 7 times, they are called 7-pass receptors. Now 7-pass receptor is an integral protein. You are understanding it? But it may be associated with some other proteins which are attached with it inside. You are understanding? This is a receptor. Receptor is an integral protein. But when receptor is activated, it gives signal to some protein which is associated inside. This protein which is associated inside, what is this? This is an example of peripheral protein. So integral proteins may be associated with peripheral protein. And this protein may produce, let's suppose, this protein may give signals further into cell, right? And activate second messenger system. Actually, let me tell you. Suppose the hormone which come here, that is the first messenger. Right? When hormone come to a cell, that is first messenger. And first messenger work with the receptor. Receptor activates some other intracellular protein. And intracellular protein produce second messenger. So in some books they say proteins related with second messenger system are also peripheral proteins. The point which I wanted to tell you, that proteins which are associated with the second messenger system, they are not integral proteins, they are peripheral proteins. But receptors are integral proteins. That's right. Any question up to this? Okay, we'll discuss further detail tomorrow.